Okay, hello everybody, and let's carry on our discussion about epidemiology and disease processes. And first of all, we need to realize that because of the cell theory, that every cell arises from an already existing cell, you'll realize that disease-causing organisms do not spontaneously arise. Instead, they have to be pre-existing in some form to then be picked up and serve as infectious agents. And the disease-causing organisms maintain their presence because they reside in what we refer to as reservoirs of infection. And these serve as continual sources of infection. So that may be a continual source of infection, or it may be a sporadic, episodic source of infection. The first thing to think about is that there are, in fact, human reservoirs of disease. Now, we're not talking here about people who are suffering an active infection, spreading it to others. That is not part of the reservoir process. Instead, human beings may act as carriers of disease. They may carry pathogens and not suffer illness themselves or have inapparent infections. And these are specifically referred to as carriers, and the most famous example is that of typhoid. Now, typhoid is a bacterial disease. It's akin to a food poisoning because it's caused by a species of salmonella. And it's an extremely serious disease. It has a high mortality rate, but people can suffer milder doses of it. And people who recover sometimes maintain a population in their gallbladder and can then go on to spread the disease, even though they themselves suffer no symptoms. So that's a typical example of a human reservoir, a carrier of disease. You might also think about diseases such as HIV, because people with HIV may maintain a population of HIV for a very long time without any apparent symptoms themselves, and yet be a source of infection to other people. Animal reservoirs are extremely important in human disease. And that is because many of our diseases are in fact zoonoses. And a zoonosis is a disease which is acquired from animals. It's transmitted from animals to humans. And even in many entirely human diseases, such as HIV, there may be an animal zoonosis origin to them. And there are any number of diseases for which this is true. And SARS-CoV-2, COVID-19, is one of them. Another is influenza, and there are many others that reside in animal reservoirs, but it can be maintained in animal populations and then transmitted to humans. And then once they're transmitted to humans, they may be transmitted human to human. The third major reservoir are non-living reservoirs. And these are especially important for opportunistic pathogens. Those are organisms which normally live in these environments, soil or water, and do not necessarily require a host to pathogenize in order to survive. Soil is one source. So, for example, organisms such as Bacillus anthracus, the cause of anthrax, that is one. Another source of disease are dust particles and things, for example, like Q fever in the American Southwest. These diseases may be transmitted by dust particles. But water is probably the greatest non-living reservoir and the most important, because so many organisms are transmitted by water and can establish populations in water. Even if they don't establish populations in the water, they may survive in water and then be taken up by a host, either animal or human. A classic example, of course, is one that I've already mentioned to you, and that is the Vibrio cholerae. Cholera is caused by a bacterium which occurs naturally in water, especially in brackish water. It periodically blooms due to ecological processes, may be picked up by human beings, where it causes a very violent diarrheal disease. And if that is the case in areas with poor sewage treatment, that may be transmitted back to the water source, amplifying the population again. And this is the cause of cholera epidemics worldwide, are especially associated with contaminated water. But the natural population is always there to seed the next bloom. You might also think about another one that I've mentioned, and that is Legionella pneumophila. Legionella pneumophila causes Legionnaire's disease. That is also a natural aquatic bacterium, and it thrives particularly in warm waters that are exposed to light. It flourishes in conjunction with a number of other organisms. And unfortunately, it grows very well in air conditioner drip pans, where it acts as a source of infection. So those are typical non-living reservoirs. Water is by far the most important transmitter of organisms for diarrheal disease. But you might, and there are any number of them that are transmitted by water. Where am I? Okay, so I gave, there is a slide there that gives you a detailed list of the various zoonoses. And we'll talk a minute about zoonoses, because apart from water, animal reservoirs are by far the most important source of both ongoing disease and new diseases that enter into the human population. Animal reservoirs act for almost all living organisms, for almost all disease-causing organisms. Viruses, which are not living, viruses, bacteria, fungi, protozoa, and various parasitic worms and things like that. The viral diseases are of particular importance to us now because of SARS-CoV-2. But over the course of human history, probably this one, influenza, has been the most important. Has been the most important viral disease by far, because it has resulted in such massive human mortalities on an ongoing basis for millennia. And influenza resides in three important reservoirs. The first are human reservoirs, but that is usually for active disease. But it also occurs in pigs and in poultry. It does cause definitely causes disease, but it's also much better tolerated, often in pigs and poultry. And they act as a continual reservoir, and as we'll hear later, they also act as a mixing vessel for different viruses, different influenza viruses, which continually create new viral species. Other viruses in animal reservoirs are rabies. Rabies is a mammal species, infects mammal species. HIV originated in primates, but later on, the HIV human immunodeficiency virus is strictly a human disease, but it has its roots, its evolutionary roots are in ape populations, which became transmitted to human beings. I've already mentioned our SARS-CoV-2. And then another disease that you've surely heard of, and that is Ebola. And Ebola is actually very well tolerated in bat populations. They don't suffer from the illness, but they release the virus in their feces, and that can be that can be picked up by human beings and cause periodic epidemics that we hear about so much. Just point out one thing to you. In terms of viruses, animal reservoirs are very important, but a particularly important reservoir are in the bats. For some reason, there's much speculation about why this is. Bats are very, very tolerant of viral infection, and they host many, 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 many different viruses without apparent harm. It's probably something to do with the fact that they are so often crowded together in very, very tight spaces. That's the one theory. Bacterial disease is also maintained in animal reservoirs, and in particular, anthrax is one of them. The bacillus can survive in soil on its own, but rich sources of the bacillus and especially of its spores arise because they cause disease in animals, especially in cattle, and those spores can then be picked up by human beings and cause disease. You can also pick up anthrax directly, by the way. You can pick up anthrax from living vegetative bacillus cells if you handle meat from an animal that is infected with anthrax. Plague is another one. Plague resides, actually, there is some controversy about this, but the classic model is that plague, which is caused by a bacterium called Yersinia pestis. Yersinia pestis infects fleas, and it is the flea that actually transmits the bacterium to human beings, usually. Once it's transmitted to human beings and they suffer from the disease, then human beings can transmit it directly to other human beings by various means. We'll hear about it when we talk about bacterial disease. But the fleas, of course, are maintained by a rodent population. And they can also pick up the bacterium, maintain it, and they appear to suffer little effect from it. And so the rodents themselves act as a reservoir, and the fleas can actually transmit the bacterium to human beings. Salmonella, birds and reptiles. This is something for those of you who keep and handle birds and reptiles as pets, whatever. Bear in mind that birds and reptiles frequently maintain populations of Salmonella, the food poisoning organism, and excrete it in their feces. And so when you're handling birds and reptiles, you need to be extremely careful not to transmit to your mouth. Make sure that you if you've been handling birds and reptiles, always wash your hands afterwards before you run any risk of picking up the bacterium. Fungal diseases are not that common, not that commonly transmitted, but there is one set. It says here ringworm, but there are actually a set of skin infections which are transmitted by animals. And there are a couple of other obscure ones that may cause serious disease, especially in immunocompromised people. There is one, for example, which is transmitted by birds, very commonly by pigeons, which causes disease in people who are suffering from AIDS. Rodents frequently act as reservoirs for protozoa, for protozoan organisms. And this is true for both of these, in fact, for both of them mentioned here, Giardia and toxoplasmosis. Rats, mice, various other rodents tolerate these organisms pretty well. Giardia causes a diarrheal disease in human beings, either acute or a chronic infection, diarrheal disease. Toxoplasmosis is an extremely interesting organism. And toxoplasmosis is a parasite It is commonly found in cats and is part of and is excreted by cats in their feces. So those of you who have cats, you need to pay careful attention to how you clean their litter boxes, etc., because it's quite easy to pick up toxoplasmosis. Usually it forms a subclinical infection, but it can form a very serious infection, especially in pregnant women. So it is something to be really careful of. I'll just tell you one interesting thing about toxoplasmosis. Toxoplasmosis is able to infect the brain and affect brain function and emotion. This is part of its kind of life cycle. It will, for example, infect rats, and the rats lose their fear of cats. And usually rats will avoid anywhere where there is cat urine. They pick up the smell of cat urine and they move away from the area. When they're infected with toxoplasmosis, they move towards it. And they actually, if they see a cat, they will actually approach the cat. You may have seen on the internet, I see them the whole time, these videos of people filming rats chasing cats instead of running away from them. It's almost certain those rats are infected with toxoplasmosis. The cat will eventually, they will be caught by a cat, eaten, and the toxoplasmosis then gets transferred to the cat, which is actually supposed to be a terminal host. But the cats can then pass it on in the feces. Lastly, the parasitic diseases such as helminths, tapeworm, roundworm, many others can reside in animal reservoirs, pigs, cattle, dogs, and very often the disease is then transmitted to humans because the eggs occur in the feces, and if we pick them up, then they're just transmitted. In addition, very often, especially with the tapeworms, the tapeworm may have a stage of its life cycle where it embeds in muscle, and we eat that muscle, eat the tissue, and acquire the helminth. So all of these are important zoonoses. But look at the slide before. You don't need to memorize it, but just have a look to see the huge range of zoonoses that occur. And on one last note, the zoonoses, these animal reservoirs of disease, are a frequent source of new and emerging diseases in human populations. And SARS-CoV-2 is just one example. So once the disease enters the human population, it has to be transmitted in order to be maintained. And there are various ways in which these disease-causing organisms can be transmitted. We divide them into two groups, the direct transmission and then indirect transmission. Direct transmission, of course, the easiest one to think about is direct human-to-human contact. And through kissing, hugging, intimate contact, sex, whatever. And this is referred to as horizontal contact. So any directly from one person to another, that is horizontal contact. And horizontal contact will also include transmission by airborne droplets. Now, this is over a relatively short distance. These droplets are not long-lived. They are large enough that they will fall out of the air eventually, but for a period of time, they can be picked up by people close by. So that is also a form of horizontal contact. Vertical contact refers to the transmission of disease from mother to child. And this there are various infectious diseases which can be transmitted to a fetus from the mother, despite the protections of the placenta, which acts as a barrier usually to most organisms. Nonetheless, there are diseases which can be transmitted from mother to infant, especially viruses. But there are some bacteria which can also move across. The last direct contact is through a vector. And we'll talk a little bit in more detail about vectors. Vectors are living animal reservoirs which are actually infected by the disease-causing organism in one way or another, and then transmitted to humans. So that is also a form of direct. It's not person to person, or it is via an animal intermediary, if you want to think of it that way. The indirect transmission, first of all, I'll say that these are kind of overlap, both of these. The first is, you've heard me use the term fomite, and a fomite is a surface on which disease-causing organisms can reside for a time and be picked up by another host. So there are numerous of them. There's certainly are bacteria which can be transmitted by fomites, but it is probably more important in viral diseases than it is in bacteria, because there are viruses which can actually survive on surfaces for quite some length of time. And the ones that spring to mind, for example, are rhinoviruses, which cause the common cold, things like that. SARS does not. It does not survive on surfaces well at all. But there are also other viruses, those causing diarrheal diseases, for example, which do survive on a fomite and can then be picked up by somebody else. The second indirect is all those important reservoirs, from those important reservoirs that I talked about before, water, those sorts of things. But food and water are extremely common sources of infection, and especially for bacteria which are transmitted by the fecal oral route. That is, when food or water are contaminated with feces from somebody infected. It is usually bacterial, but it can be viral as well. That acts as a vector, if you want to think about it that way, but not in a strict biological sense. But the population of bacteria may be maintained in food, or it may be amplified in food or water, and then picked up by humans, and then act to cause disease. And in the case, especially of diarrheal diseases, it is very easy for people then to also infect fomites and spread it even further. So these two do overlap. The last indirect means of transmission is airborne. Now, this is different to this direct horizontal by airborne droplets. This refers to very long-lived transmission through the air. One of the diseases in which this occurs is tuberculosis. The bacteria, although they're not spore formers, they are very tough bacteria, and they will survive for a long time in the air. And so that would be regarded as a droplet model, but not as a direct necessarily a direct transmission. The last I'll mention again is dust. Not terribly common, but there are some distinct diseases, which we'll hear about when we talk about disease itself, which are transmitted by dust particles. Okay, so let's have a look now at this, the direct transmission of disease by vectors. And a vector is strictly defined as a living thing that moves a disease organism to a host. So the most important group of all are arthropods. There are some other vectors as well, but arthropods are by far the most important. And there are three groups, two of them insects, one of them an arachnid. Fleas and mosquitoes are both insects which transmit disease. And ticks are another group, a very large and diverse group of arachnids related to spiders and everything like that, which transmit many, many other different diseases. And they transmit diseases of disease-causing organisms of many different kinds, viruses, rickettsias, parasites, or bacteria, all sorts of things. So the first kind of transmission is simple mechanical transmission. And then we immediately think about flies. Okay, that flies may land on feces, they may land on dead animals, and then land on your food and transmit disease. Well, that is true, but it's actually not nearly as common as one might imagine. Flies do not actually transmit that disease that way very frequently. There are other diseases, however, that they do transmit. They certainly can cause food poisoning, but you would have to be very, very unlucky to acquire a food poisoning from flies landing on your food, to be honest. But there is another disease. There are other diseases, and I'll mention just one as an example. And this is called trachoma. It's actually caused by a Chlamydia trachomatis, a bacterium, a very strange bacterium, but a bacterium nonetheless. And that can cause a disease of the eyes, and that is transmitted by flies. And in tropical areas, especially of Africa, it has been a major problem. It is now less of a problem than it used to be. Still a problem, though. The most significant vector transport, however, is not mechanical. Instead, it is biological transmission. And this is a situation where the vector themselves become infected with the disease-causing agent. The disease-causing agent undergoes at least a part of its life cycle in the vector. This is true, for example, of mosquitoes picking up malaria. The malaria actually breeds in the mosquito and is then transmitted to a human host. There are many, many of these diseases for which this is true. And these are probably the most important ones that you need to remember for the moment. I wish I could just get rid of these bars entirely. Maybe you can't see them, but I can. The transmission of the disease-causing organ from the vector to the host can be by a variety of means. Most importantly, bites. That would be the case for mosquitoes biting somebody if they are infected with some disease-causing agent, malaria, West Nile virus, something like this. They transmit it to the host via the bite. But there are others which are transmitted a little bit differently, and plague is one of them. Plague is transmitted by fleas, but it's not injected into the host. Instead, it is laid down on the skin of the host around a flea bite, and people scratch themselves and infect the scratch with the disease-causing organism. A similar thing works for another disease called typhus, which is not to be confused with typhoid fever. Typhus is an entirely different disease, also transmitted by not by fleas, but by lice, which is another insect. Okay, so I've given you here, just 1 second. I've just got to move my bars. I've given you here a slide with different arthropod vectors and the diseases that they transmit, and you'll see that there's a huge variety of different diseases which are transmitted by these vectors. Okay, well, now what? Sorry. Okay, so I just want to finish off with a very brief discussion of healthcare associated infections for you who are interested in careers in nursing, medicine, or whatever. This is a very important topic, and I'm sorry that I've just got to skip over it, gloss over the details. But I just feel it's so important that I have set as an extra credit, 1% extra credit. Please review the slides that I'm going to discuss now. Prepare a 2-page maximum printed explanation, just with a brief introduction about what these infections are and how they arise, but devoting your attention instead in the writer to an explanation of effective methods to prevent these infections. Now, healthcare-associated infections are referred to as being nosocomial infections. Now, they are specifically associated with a healthcare setting, and especially hospitals. And we need to realize, of course, that these are, in fact, extremely common. And there are various reasons why that is the case. In fact, about 1 in 25 hospital patients will acquire a nosocomial infection. And that applies also to people reporting to emergency rooms, etc. In the United States, about 2 million people a year are infected. And shockingly enough, there are over 70,000 deaths associated with nosocomial infections. And 30% of those infections are entirely preventable by relatively simple means, as we'll discuss. So we might ask why. Well, actually, the reasons why people are particularly at risk in hospitals are fairly obvious. First of all, you're crowding people together in an enclosed setting. That is, they're close by one another. There are people moving around close by them the whole time. And there can be resident microorganisms in the hospital environment. In droplets, air droplets, as spores, various other things. The second thing is, of course, many people in hospitals, unsurprisingly, many people are ill, and they have a weakened immune system. And as a result, they are more susceptible to infections. The third thing is that, unfortunately, the miracle of antibiotics, which heralded the new age of healthcare, of curing so many diseases that had formerly been so serious or lethal, that has resulted in the overuse of antibiotics and the development now of resistant strains. And those resistant strains commonly occur in healthcare settings, because we have people returning to the hospital regularly because their infections have not been cured by the antibiotics they were previously given. And so they themselves are a reservoir of antibiotic-resistant strains of bacteria. And the fourth thing is that there is a chain of transmission. In fact, the healthcare workers act as vectors. They actually pick up disease-causing organisms, either mechanically on their hands, etc., or even as infections in themselves. And people may, for example, acquire a respiratory infection from a patient and yet carry on working. That's an example. Come back to work day after day after day while they themselves are ill and spread it to other patients. There's one group of patients who are especially at risk, And this is where most of the mortality occurs from nosocomial infections. And these are people who are compromised. That is, they are immune compromised. And these are people whose resistance to infection has been drastically lowered. Serious burn cases are one. People who have immune diseases such as HIV and other diseases of the immune system. People who are undergoing immunotherapy, chemotherapy, all of these things may affect people's immune status and make them much, much more liable, even to organisms which normally only cause mild or no illness at all. So here are the principal sites of healthcare-associated infections. First of all, urinary tract infections, and we'll hear about why that is. Second, surgical site infections. Although we are used to thinking that we have entirely evaded the problem of surgical infections, that is not true. Surgical infections remain relatively common and certainly are one of the commoner nosocomial infections. Then respiratory tract infections, especially lower respiratory tract. Upper respiratory tract infections are remarkable only in that they may predispose to other infections. But lower respiratory tract infections are extremely dangerous because they very frequently lead to pneumonias in hospital patients. First of all, because hospital patients are somewhat compromised. Second of all, because they very often are immobile for much of the time. And pneumonias become extremely important as for morbidity and mortality. And then various others. The gastrointestinal, this is of particular interest because this does relate to the overuse of antibiotics in this particular case, and that is this infection, Clostridium difficile. Clostridium difficile is an organism which causes a very, very serious gut infection in patients who have lost their normal microbiota due to the use of antibiotics. We'll hear about it later. I won't discuss details now. Then various blood stream infections and other. So there are a wide range of different things which can arise in a hospital setting. Surprisingly enough, the most common source of problems is Staphylococcus aureus, which we've heard about so much and you will have seen in lab so frequently. Staphylococcus aureus, the gram-positive coccus. It is a normal part of the skin microflora and the hair and the outside of the body, where it causes very little problems. But once it gains entry into the interior of the body, it can cause very, very serious problems. And it is the major source, for example, of postpuerperal infections, that is after childbirth. It's also the most important cause of post-surgical infections. And that for various reasons, of course. First of all, personnel are a source of Staphylococcus aureus. It's on their skin, it's on skin flakes coming off healthcare workers. So that we all are a source, constant source of Staph aureus. There are various others that also are by and large tolerated in our microbiota and or significant parts of our microbiota, which placed in the wrong setting become opportunistic pathogens. And one of them is Escherichia coli. The other is Enterococcus. Both of these are members of our gut microflora. And the last one is Candida. Candida is not a bacterium, it's a yeast. There are all sorts of ways in which these organisms, by the way, this is just a very, 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 very short list of the kinds of organisms that can be transmitted. But there are various ways in hospital settings in which a patient is placed at particular risk of acquiring infection. The first, and probably one of the most important, is through the use of catheters. Urinary catheters, especially if they are indwelling, they left there for a long time, are a major source of urinary tract infections. And those urinary tract infections, not just urethra and bladder, it may actually extend to kidney infections as well and be extremely serious and life-threatening. Surgical procedures, fairly obviously, once you open that sacred interior sterile part of the body, which comprises the interior of the abdomen, the interior of the thorax, the interior of the head, whatever, any bacteria which get in there violate that sterility and can breed very, very quickly and cause massive problems. So surgical procedures still remain a major source of infection. There are, in addition to that, in a way related to it, are indwelling ports and venous catheters and things like this. I say related to the surgical procedures because they are providing a gateway from the outside, highly non-sterile, to the interior, which is supposed to be absolutely sterile. And this can be taken advantage of even by organisms which normally cause very few problems, such as Staph aureus, even Staph epidermidis. The last one, but of particular significance of the last 2 years, has been mechanical ventilation as a source of nosocomial infection. And this is very, very difficult problem because this mechanical ventilation is pushing air at pressure down deep into the lungs. And it is bypassing the mucociliary escalator, which normally keeps the lungs and the lower respiratory tract almost sterile. There is a possibility that you can push bacteria deep into the lungs, and especially, for example, in a respiratory unit where people may already be suffering from bacterial pneumonias, this can actually be easily done. And it is a frequent cause of mortality and of morbidity, of course. But it has a high mortality in people who are already so compromised that they are on respiratory therapy. So this is what I want you to pay particular attention to. Many of these infections are caused by healthcare workers and by careless methodology, put it that way. The standard precautions, the standard common sense, well-established hygienic precautions. First of all, hand washing. That is one of the most important factors in the transmission of disease through a hospital setting. And yet, believe it or not, surveys have shown that healthcare workers can be extremely, extremely careless about hand washing and about basic hygiene. Then attention needs to be paid to all of the transmission, to all of the other ways in which transmission could occur. And first of all, there might be a necessity for protective equipment. There might be a requirement for a special housing for patients who are suffering from highly infectious diseases, so that there's no chance that they can transmit to other patients, etc., etc. Go to your text and read through this carefully and include that in your write-up. Okay, so I'm going to leave it there. And we're now going to move on and just be able to just give you a bit of an introduction to our discussion of viruses. And the viruses are the first group that we are going to, of disease-causing organisms that we're going to discuss. First of all, of course, because they are so tremendously important. They're hugely important in the number of diseases that they cause in human and animal populations. And also because they are major contributors to human mortality and have been for all of the millennia. So some general characteristics of the viruses. The first thing to state very firmly at the beginning so that everybody has it clear in their minds, viruses are not alive. They are non-living infectious particles. And what they do is they will co-opt a living cell. They absolutely require a living host in order to multiply. There are no free living viruses. All viruses are obligate intracellular parasites, and they require a living cell to manufacture all of the viral components. They are unable to manufacture their own viral components to reproduce themselves. So all of the components are manufactured by the host and then put together and released in one way or another, which we will discuss. Here are some reasons why they were not considered living. Okay, they do not have any of the mechanisms for metabolism. And especially, they do not necessarily have DNA as the hereditary material. Remember that that is an absolute requirement for our definition of life. All living things depend on DNA because it's transcribed to messenger RNA and translated to protein. But viruses do not necessarily have DNA. They may have RNA as their genome. So there are DNA viruses, but there are also many RNA viruses. At a very minimum, they have a genome, the hereditary material, which is going to be used by the host cell to produce new viral particles. And that is protected by a protein coat. That's the very minimum. So that's our basis, and we may elaborate on that. Although within that protein coat, they may contain some other proteins. Sometimes they have their own polymerases. And sometimes they have their own proteases. A protease is enzyme which cuts proteins into smaller proteins. It cuts a long polypeptide into functional protein units. They lack ribosomes, another reason they are not alive. They have no means of metabolism. They have no way, for example, to generate ATP. They don't generate ATP because they don't need it. They have no cytoplasm, and they have no cellular machinery that needs to be maintained. They are inert, entirely inert. That mature functional virus particle is called a virion. I noticed, by the way, that this line is missing on your printout, on your PowerPoint. So please write that in. A mature functional virus particle, the equivalent of a cell, but nowhere near it, is called a virion. Very often just referred to as a viral particle as well. They're extremely small. So let's just for comparison, here's a red blood cell, a human red blood cell. And the red blood cell, the plasma membrane of a red blood cell is 10 nanometers thick. That is a millionth of a centimeter, right? And so it's extremely, extremely thin. Most viruses, if you look at this list, are between twice that, 24 nanometers, and leaving these ones aside for a minute, and up to about 250, so it's 25 times, between 2 and 25 times the thickness of the cell membrane. Very, 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 very small indeed, far below the ability of a microscope to see them. There are some larger viral particles. One of them here, this is the vaccinia virus. This is an artificial virus. It was created to act as a vaccine for smallpox. And it's a very large particle. It's a living particle, but it's extremely large. And then there are some which are very, very long, but thin. And this is a bacteriophage, a virus that infects bacteria. This is the famous Ebola virus. And it is very long for a viral particle, and also, but much thinner than it is long. But that is probably one of the largest of all of the, excuse me, all of the viruses. I'll just mention, by the way, there are some extremely small bacteria as well. And the two groups which we look at with real interest is this group, the chlamydia. Here you can see it's only 300 nanometers. It's almost the size of the virus. And then the other group are the rickettsias, which are also extremely small. And some people believe that, in fact, the viruses arose from one of these very, very, very small bacteria that eventually lost all of its cellular machinery except for its genome and became an infectious particle. That's just for interest. Okay, so a characteristic of the viruses is that they tend to be very, very specific. In fact, some of them are extremely specific. And they are limited to a specific host or group of hosts. And in that group of hosts, they are restricted to particular cells which they can infect. So I have to tell you that everything that is alive has its viruses. Bacteria have their own viruses called the bacteriophages, and there are thousands of them. Algae have their own viruses. Plants have their viruses. Of course, animals have their viruses. Anything that is alive, you can guarantee it has its viruses. In fact, there are viruses which need to infect another virus in order to infect a host. So that is how diverse they are, but how specialized they also are. It's very common to find viruses that only infect one particular host and only one group of cells within that host. But others will infect a range of hosts. So, for example, the rabies virus will infect mammals. Any mammal can be infected by rabies. Some of them tolerate it far better than others. And there are some groups which almost are never infected. But rabies potentially can infect any mammal, nothing else. A disease like measles is restricted, for example, to the apes, including us. So any of the apes can suffer from measles. HIV, on the other hand, is highly specialized. It's specialized for human beings only. And not only that, it is specialized for particular cells in the human body. It only infects those cells which carry particular receptors on their surface. And those are largely, not entirely, but largely members of the immune system. And we'll mention later on that that's one particular cell that has its major effect on human health when it is infected with HIV. So many of the viruses, I've mentioned this already, but please bear in mind that many of our viruses arise as zoonotic viruses, the cross from animal to human. They establish and they diversify in animal populations. and then cross over into the human population. And the ability to do so depends upon them being able to bind to specific receptors on cell surfaces. That is how they recognize both their host and the tissue to which they are supposed to attach. They only bind to cells which have the correct receptors for that particular virus. So let's have a look at the actual structures. Here is a relatively simple virus. And this is a polyhedral virus. It has this geometric shape with the flat sides. This is an outside shell of protein, and this is called the capsid. This is a feature of all viruses. They all have a capsid, and the capsid encloses the genome. The capsid is made up of these protein subunits, these repeating subunits, and each one is called a capsomere. On the inside is the nucleic acid. And as I've told you, that nucleic acid can either be DNA or it can be RNA. It depends on the virus that you are talking about. This is a very common virus shape. There's one important virus for us that has the shape, and this is adenovirus. And adenovirus is a common human virus. Viruses may also have a helical shape to the capsid. This is the very first virus that was ever visualized, but it's also the first virus that was ever described, and it was described long, long, long before it was visualized. This straight form like this is tobacco mosaic virus. But there is a famous virus which also has this helical shape, which you will have heard of, and this is Ebola. And Ebola is easy to recognize. It's a long filament. It's very long for a virus. And at the end, it has this kind of squiggly shape that curls back on itself, and it's often referred to as a carpet beater shape. If you don't know what a carpet beater is, you'll have to go Google it. I'm not going to explain it. It's self-explanatory. But that's typical for Ebola. But you'll see this part here is exactly that. It's a helical virus. Here are the capsomeres stacked in a spiral, and then the nucleic acid is wound around on the inside of that. But viruses also occur which have around them a membrane. And that membrane is derived from the host. So here is a virus. This virus has a polyhedral capsid on the inside. Here's the genome here. But on the outside here is a membrane. And that membrane can be derived from various sources in the host. It can be derived, it depends on the virus. It depends on the life cycle of the virus and how it is produced inside the host cell. But it can be produced from endoplasmic reticulum membrane, from nuclear membrane, from Golgi apparatus membrane, or from the cell membrane. And we'll see how, when it's invested with the cell membrane, we'll see how that occurs in a while. But it is a coat around very, very similar in structure to the host membranes, except for one thing, and that is it is embedded with viral proteins. You can see the viral proteins all the way around, appearing as spikes. And those of you who know anything about the COVID virus or about HIV will recognize those because they're characteristic of the viruses. These spikes on the outside are what enable the virus to determine the correct cell to bind to. And presence of those spikes with receptors for them on the cell enables the virus to bind to the host cell and then to enter the host cell. So that capsid there on the interior, it can be polyhedral like that, or it can also be helical. like this. And this is really pretty much what a COVID virus looks like. It has a helical filament for the capsid on the inside, with an R, in the case of coronavirus, an RNA genome on the inside of that, surrounded by a membrane which is derived from the host. So when they just have the capsid only, we say they are non-enveloped, or it might be easier to remember as non-enveloped. Okay, we'll do it that way, non-enveloped, even though proper pronunciation of this word would be non-enveloped. They are non-enveloped. They have no envelope around them. These ones do. They are enveloped. And that envelope is derived from the host membranes. There is a third group, and this is a group, I confess, always just amaze me. These are called the complex viruses, and these are typical of viruses that infect bacteria, and these are the bacteriophages. Not all bacteriophages have this shape, but there are large groups of them that do, common groups that do. And I'll just finish off by explaining a little bit about this function. First of all, this is the capsid here at the top, the polyhedral capsid. And on the inside is the nucleic acid. In this case, it's DNA for this particular virus. This is called a T-even bacteriophage, and it has a DNA genome. Below it, there is a sheath, which is made up of spiral protein. And under normal circumstances, that spiral sheath is like a spring, and it is extended. On the inside here, there is a tube that runs from the capsid down. These here on the bottom are they're called tail fibers. They're like legs of a spaceship. Those are the equivalent of viral spikes, which determine the correct cells to bind to, because these are very specific to certain bacteria. So they will only bind to certain bacteria, and they recognize features on the outside of the bacterium and bind to them. When this little mechanism binds to the outside of the bacterium, what it will do is the spring will collapse, and it pulls down this spike, and it pushes the spike right through the cell wall of the bacterium, through the cell membrane, and it forms a pore through the cell membrane. And the genome is then injected into the host. The whole thing, all of this, remains on the outside. Here you can see here, these are two discharged. This is one undischarged. It hasn't spat its genome in yet. And so these are viral particles sitting on the outside, but the genome is injected into the cytoplasm of the host to then be replicated. And we'll learn something more about their replication when we meet again on Monday. Remember, please, your third test is not on Monday. It will be the following Monday, the Monday before Thanksgiving. And the test is going to be entirely on DNA and DNA function. And I've posted review sheets for you to look at. Okay? So I'll see you then on Monday.